Morning. Title of my presentation is A National Perspective on HABs, Moving from Monitoring and Mitigation to Prevention and Remediation. It's really divided into about four components. I want to provide some historical background to how we came to our current understanding of cyanobacteria harmful algal blooms as opposed to the more common marine harmful algal blooms like the red tides. Some of the important researches that led to our current understanding, and then move to a better understanding of how cyanobacteria come to dominate in our water bodies, how that understanding of that can inform us using methodologies that currently exist for management and mitigation, but using those in a more scientific pattern and by using and informing ourselves with something called systems approach, and utilizing those approaches to understand how cyanobacteria dominate, and then use examples. Cyanohabs. It's been about 50 years, even though cyanobacteria have been around for a couple billion years, but it's been about 50 years since the term eutrophication has been definitively linked to cyanobacteria blooms and has entered the common language. In 1974, cyanobacteria were recognized as producers of toxins, in particular the neurotoxin anatoxin a potent depolarizing neuromuscular blocking agent that was the topic of my PhD thesis. In 1998, we had the name and general structure for the peptide liver toxins called microcystins, named after the genus Microcystis. And it turns out that these toxins are really the more prevalent worldwide and the ones that are more responsible for water quality issues and human and animal mortalities. In 1990s, we had developed a very rapid, fairly rapid, easy to do test for microcystins called ELISA, enzyme-linked immunosorbent assay. That assay methodology is still used worldwide now to detect microcystins in water supplies and even in animal tissues. In 1998, the Harmful Algal Bloom and Hypoxia Research and Control Act led to the establishment of a federal interagency working group led by NOAA and the US EPA. By 2005, we had about 75 different cyanotoxins identified structurally. Most of those were microcystins because, as it turns out, microcystins compose many different analogs of a pentapeptide, and all you have to do is substitute different amino acids and you have a different microcystin. By 2015, there were about 300 different cyanotoxins. Again, many of those were microcystins. There's well over 200 microcystins. Fortunately, those microcystins can be largely detected by that same immunosorbent assay methodology. In 2014, the whole understanding of microcystins and their impact on water quality and animal and human health came to a head in Toledo, Ohio, the western basin of Lake Erie, where a massive bloom of microcystis and microcystins, which then infiltrated into the finished potable water and was in the taps of people's water, led to a cancellation of water supply for about 500,000 people for 5 days in the area of Toledo, Ohio. 2020, the U.S. Government Accountability Office was tasked to investigate and report on cyanohabs. That report came out in 2022, and I want to return to that report and do some examination of that report and where we are with the recommendations of that report. But first, the bloom in Lake Erie, which sort of brought this to a head, was about 700 square mile bloom of toxic microcystis. But along those same lines, other places were experiencing blooms, including here in Oregon. Our water supply for the capital city of Salem was shut down. West Palm Beach, water distribution problems, and of course, top right, that bloom in Ohio. So the question to be asked is, following that water quality crisis in 2014, what has changed and what hasn't? Well, what has changed, to the positive is that we do have much better methods for detection, monitoring, identification, genetic methods, chromatography methods, the ELISA methods, remote sensing methods. There are many, many of these that have been developed and continue to be developed and improved upon. But what hasn't changed is we still have lots of blooms. The next few slides sort of illustrate some of those blooms internationally. First one being the one here in Oregon, drinking water. This is the bloom on Detroit Lake Reservoir in 2018. Notice that it's not your typical blue-green, but a brownish orange. This was an anabina, or more properly now called Dolichospermum, producing microcystins and anatoxin A. In Ireland, the bloom looked like this. In England, Lake Windermere. Lake Windermere has been the classic lake studied for centuries to define methodologies for limnology, but it also can experience toxic blue-greens. In Pakistan, the water supplies in many cases have been deemed unsafe. In Brazil, which has had issues for decades now, and also has the examples of human fatalities from utilizing microcystins in water supplies in a dialysis center, which killed over 100 people. In Australia, Australia has a unique position similar to South Africa in that they have periods of low rainfall, and so they store big water supplies in reservoirs that are quite large and shallow, and without any flushing or removal, they create a phenomenal situation for blooms to develop. In South Africa, which this slide shows, this is a slide showing water hyacinth, not a cyanobacteria bloom, but Harpiesport Dam for decades has had issues because of high nutrient inputs and massive sediment accumulations from raw sewage water. Initially, it was macrophytes like these water hyacinths, but they herbicided the water hyacinths to remove them so that it could go back to recreational use. And in the dumping of that nutrient into the water supply and the opening up of the surface waters allowed massive blooms of microcystis to become dominant and microcystins. So in more recent times, they've kind of allowed the lake to resort back to water hyacinth, but it hasn't in any way, shape, or form improved upon the water quality. So news reports of HABs from about 2010 to 2019 show this sort of steady increase. It's largely through increased monitoring, but also the increasing levels of phosphorus and nitrogen, and along with increased temperatures, along with global warming, and harmful algal bloom events have grown about 7-fold. And this is a conservative number in reality. So these HABs have substantial direct and indirect economic health effects, drivers of increased attention and regulation around nutrients, especially agriculture, so phosphorus and nitrogen. The economic costs have been estimated in a few cases, but certainly not to a great degree. So in Florida, the estimated yearly HAB losses of about 33 million, 27 million in commercial fishing. West Coast blooms of pseudonytzia, which is really a diatom, not a cyanobacteria, but still it's a harmful algal bloom, costing the Dungeness crab industry about 50 million a year. And that toxin is called domoic acid. It's actually a peptide, again, that crosses the blood-brain barrier and leads to amnesia and even death. The recent HABs occurring in the southwest corner of Lake Erie are estimated to exact about an economic cost of about 70 million. So the current approaches involve what I refer to as the 3 M's. They involve monitoring. Monitoring really means getting good at detecting what's there. So it involves remote observation, be satellites or drones, more sampling directly of the water body, better methods of identifying the organisms responsible, so taxonomy, and then of course cyanotoxin detection itself. All of these processes inform how you set HAB advisory guidance, management, and mitigation, but it doesn't typically address prevention and remediation. Management and mitigation, you have to keep in mind, is an attempt to reduce, control cyanobacteria and their HABs. And these approaches serve mainly to minimize blooms. They don't affect prevention. Now, true prevention really means going after better water quality, and often that means improving the watershed. But it's proven very difficult to control watershed runoffs because of all the development, commercial, residential, et cetera, farming, that really, if you take it out of that use, then you no longer have it for those functions. So what that really leads to is that you're forced to start to affect prevention and remediation, coming from remediation inside the water body. And that's my emphasis for this talk. So lake reservoir management and mitigation tools really come down to these three following. Physical controls, that's often what's used in water treatment plants for potable water. You manipulate the intake of the depth. You put in aerators for destratification, mechanical mixers to circulate, and even barriers of sand and carbon. Biological controls really come down to how do you manipulate that lake ecology to favor the grazers, reduce the cyanobacteria, sort of a top-down effect, and increase competition for nutrient utilization by other organisms, not just cyanobacteria or a bottom-up control. What typically, though, is used in management and attempts at mitigation are chemicals. Often these chemicals focus on the key nutrients, phosphorus and nitrogen. In the case of phosphorus treatments, the idea is to remove the effect of that phosphorus. You don't really remove the phosphorus itself, but you use lime, aluminum sulfate, ferric chloride, lanthanum, which is commercially called Phoslock, and clay particles. These bind the nutrients, flocculate the cells, and of course, try and take that phosphorus out of the system, at least bind it up so it's not available to nutrient cycling. But also used are algicides, the old standbys like copper sulfate, hydrogen peroxide, potassium manganate, and even herbicides directly. But keep in mind, all these are temporary, and we want to illustrate why we can do better. These three examples are used widely in reports and methodologies, but in this case, this comes from a UNESCO report called Solutions for Managing Blooms. Let's go back to that GAO, the report that I mentioned earlier. Coming out of that report were lots of recommendations, but what's happened to those recommendations? Well, that working group has not implemented a national HAB and hypoxia program. The working group has not developed performance measures to assess the results. What they have done is help state, local, and tribal governments respond to HAB events, hypoxic events, but lacks prevention. Those federal agencies have identified a need for more actions to prevent HABs and hypoxia. And they have developed limited cost and benefit information. And that information is being used to select amongst the mitigation methods, but only mentioning the need for prevention. Some factors that contribute to harmful algal blooms and hypoxia. This cartoon will be used to illustrate in more than one situation how we can move on. We recognize that land use provides excessive phosphorus and nitrogen runoff. That runoff leads to algal bloom formation. Algal blooms decompose, consume oxygen. That decomposition product settles into the sediments. Those sediments build up. The low oxygen conditions in those sediments lead to animal mortality. But because they're hypoxic, that sediment releases the nutrients, especially the phosphorus, and creates higher levels at the low oxygen zone. And as you can see, it sets up a situation almost of recycling. But along with that, we're going to affect the food web, especially invertebrates and fish. Those get diminished or completely removed from the system. So leaving that cartoon up, what we have in effect done with our land use practices and water use practices is affect an ecosystem habitat change. That change is biological. It really involves just decomposition and hypoxia and organic buildup, nutrient cycling instead of nutrient removal. So it's biological, not physical, to a great degree. It also involves feedback loops and tipping points. In other words, we have a complex adaptive system which can be informed if we embrace and utilize something called systems theory. Systems theory can help explain the process how HABs become dominant or cyanobacteria become dominant as well. In systems theory, we have attractors that describe the main determinants of the system. In a healthy lake, those attractors are an oxygen-rich environment, productive, diverse food web, and nutrient clearance from the aquatic system. For cyanobacteria to dominate, the attractors obviously need to be changed to an anoxic benthic environment, constrained food web, and nutrient recycling instead of nutrient clearance. It certainly involves feedback and tipping points. So in 2015, a group headed by Cottingham Lab used that systems theory paradigm to describe and model cyanobacteria's ability to transform low nutrient lakes into a eutrophic state in which they bloom and dominate. And in describing that system, as you can imagine, the process is complex, and it's really too complex to be countered with simple chemical treatments or single management methods that can lower nutrient levels like binding phosphorus or killing the phytoplankton, but it doesn't affect long-term remediation or prevention. The factors that contribute to HABs and hypoxia, and this again is detailed in the GAO report, even though they don't really affect methods for prevention. The critical factor is hypoxia and decomposing organic sediments causing hypoxia. Hypoxia constrains the food web and drives nutrient cycling. It creates these feedback loops, which we can sort of summarize as algal blooms, decomposition, hypoxia, nutrient recycling, and algal blooms. That progression to cyanobacterial HABs is referred to as ecosystem habitat change. And as you can imagine, that emphasizes the need for proactive, prevention-oriented approaches. And GAO admits they recommend that. They document and define a national HAB and hypoxia program. They recommend developing those performance measures, an interagency framework for monitoring and forecasting that prioritizes water bodies for prevention and remediation, developing a national goal focused on preventing HABs and hypoxia, and a more comprehensive body of information on the costs and benefits. Costs and benefits inform the better methods you can use for mitigation and control and prevention. Condense that back down, develop a national HAB hypoxia program, performance measures, monitor forecasting, prioritizing, and focusing on costs and benefits for prevention. What about prevention and remediation? Well, using that cartoon previously, the kinds of things that we recognize and want to focus on then is how to affect some sort of decomposition. How do we take care of the oxygen problem? So in other words, how do we decompose those sediments that have low oxygen? And how do we prevent then hypoxia? Next, we want to affect nutrient recycling instead of, nutrient removal instead of nutrient recycling. And then we want to address food web distribution. So really 4 things: directly affect and remediate, prevent hypoxia, then how to improve decomposition and control that decomposition so we get nutrient recycling, and then that allows a restoration of food web disruption. Now, cyanobacteria's competitive advantages, we know they've been around a long time. They're one of the first life forms to photosynthesize. They are highly adaptable. Clearly, they're adaptable if they're still with us in such, complex situations. By retaining their abilities that they've developed over the millennia, they exploit nutrient availability in eutrophic environments at the benthic margin, allowing them to outcompete and dominate other phytoplankton. Doing this, they have some tools. A key tool is nitrogen assimilation. They have the ability to fix atmospheric nitrogen. through specialized cells. So they can actually use dissolved nitrogen gas in particular. They also can go and use ammonia supplies much more efficiently than other phytoplankton, especially at the hypobenthic margins. And they have buoyancy control. They control their buoyancy through gas vacuoles, little gas bags that allow them to dive to the bottom and load up on nutrients or rise to the top and take advantage of optimal light. They also, by dropping down to the sediment and nutrient boundaries, they can mobilize sediment nutrient stockpiles more efficiently. They actively mobilize nutrients in the sediment, often by the use of extracellular microbiome aids. In other words, they have bacteria attached to the cyanobacteria cells, and those bacteria affect better absorption of nutrients, not just the presence of them. And of course, what has been driving lots of the research is the toxin production, the recognition that these organisms, especially the planktonic ones, produce very potent toxins that affect animal and human health and certainly affect drinking water supply and use and even recreational use. Now, we don't really know what these toxins are used specifically by the cyanobacteria for, but there is some evidence that it does make them more unpalatable and therefore reduces the grazer's ability to eat them, and they may be used as defense mechanisms as well. But cyanobacteria utilize these tactics to exploit the eutrophic conditions. In other words, they affect ecosystem habitat change and come to dominate. Summarizing, the root causes that drive this process are organic sediment accumulation, which leads to decomposition, which leads to hypoxia, which leads to recycling of the nutrients instead of nutrient removal, especially those anaerobic benthic margins. And the cyanobacteria can take advantage of that by diving and dominating nutrient uptake. The hypoxia diminishes or even removes the food web, especially the zooplankton and even upwards to fish. Nutrient clearance capacity of the food web is constrained. Nutrient recycling replaces nutrient clearance. Internal nutrient recycling becomes self-sustaining. Cyanobacteria exploit the ecosystem habitat change. Putting that all together, what do we want to do? Eliminate hypoxia, deplete sediment nutrient stockpiles, restore phytoplankton balance. In other words, we'd like to see a lot more beneficial algae, especially diatoms and green algae, and restore the food web to restore nutrient clearance channels. In other words, getting those nutrients out of the system rather than just recycling them. To do that, I have several examples. I'm going to rely on a company that's across the river from New Jersey, Philadelphia, called SIS Bio, and I've watched the progress of their system affect prevention and remediation since about 2017. So I want to use several examples from them to illustrate what I've just outlined as how can we address hypoxia, decomposition, nutrient cycling, reestablishment of better food webs. And to do that, let's start with the first of these. How do we affect hypoxia and oxygen levels? They have a system that's referred to as radar. It is oxygenation. It does not use oxygen. It uses air, but it's in the tire water column. And in it, what it does is place specially designed microporous aerators into the sediments, and they're placed in a situation where they're able to affect the lake by studying the morphometry of the lake and the better placement of those systems. They are different to aeration. In other words, just placing bubblers into the system where those bubblers are on the bottom or whether they're somewhere in the water column, because aeration tends to typically just stir it up. A good example of that is Lake Carmi in Vermont, in which an aeration system, just a bubbler system, was installed to try and mitigate cyanobacteria blooms, and it had unintended consequences of simply mixing the phosphorus-rich bottom, and that led to the higher levels of phosphorus and stronger, more protracted cyanobacteria, forcing them to completely remove that system and not being able to remediate or prevent cyanobacteria blooms. So let's look more closely then at this different aeration methodology called radar. Secondly, we want to use examples that affect how that sediment is decomposed and released. And to do that, keep in mind that sediments do have native bacteria and fungi, which would decompose those organic sediments, but they do that at a rate that's maybe unacceptable to see some fairly good quick responses. So this company has a system referred to as enzymatic biodredging by adding enzymes to those organic sediments. These are not microbes, but they are enzymes. And the third thing to do, keep in mind that once you improve that digestion and release those nutrients, you want to affect something that allows those nutrients to be utilized by beneficial algae. So there is a third component called micronutrient supplementation. It's a bioaugmentation to boost those levels of the food web, improving the growth of diatoms and green algae in particular, as opposed to cyanobacteria. That enzyme mix is a enzyme mixture referred to as composing of mercaptases and amylases and proteases, and the micronutrient mix of the micronutrients like iron and manganese and cobalt. So these three methods combined allow this company to show some fairly effective remediation prevention in lakes and even reservoirs. So I'm going to use these three methods and show you examples that utilize these methods for the rest of this talk. So let's look at eliminating hypoxia. The example is Indian Lake, Missouri, average depth of 15 feet, 320 acres. There was high levels of eutrophication for several years. The tipping point was reached in summer of 2020, escalating the need for responsible decision-making. And by 2021, the lake experienced high levels of toxic cyanobacteria all summer. The remediation program was started in 2022. So initially, I'm just going to show you the results of the aeration and the hypoxia effect. So in 2021, the oxygen profile of that lake looked like this. Across the bottom, depth in feet, oxygen levels acceptable at the surface, but totally unacceptable at depth. Along with that, in those anoxic sediments was the release of phosphorus, and those phosphorus levels went up to levels that supported those cyanobacteria blooms. In 2023, after effecting the hypoxia treatment, the aeration treatment, oxygen levels were restored and phosphorus levels were reduced. Along with that, there was ammonia effect. Here, this is the ammonia graph on the left, 2020-21, the maroon graph showing the increase in ammonia. And by 2023, the improvement in the oxygen and the lowering of the ammonia. So that affected the hypoxia. That was, in effect, controlling hypoxia. Well, next step is not just enough to control hypoxia and get the oxygen and nutrient levels down, but how do you deplete those sediment nutrient stockpiles so that the problem doesn't continue when the next time summer comes around? So how do you deplete those sediment nutrient stockpiles? I want to move from this lake in Missouri to a lake in Virginia, smaller lake, 30 acres, 5-foot average depth, maximum depth about 17. It was an irrigation source, and it was also a recreational purpose, but because there was a reduction in the volume, so the storage capacity to sediment loss due to sediment accumulation, and a heavy weed growth that reduced recreational use. The program began in April of 2018. So in 2017, this was the depth profile, the lighter colors showing the very shallow margins all the way around the lake, only the bluish colors showing the depth, which was the more open areas. So if we take a transect across that lake, flip it on its side, showing the depth profile in purple, notice that over half of that lake had depth profiles of only 2 to 4 feet, and there's only depth utilizable for irrigation was really the other half of that lake. So back to that depth profile look. The other issue that was a real problem with this lake was macrophytes. And initially, this is what the 10-foot contour line looks like in 2017, keep in mind. But the macrophyte profile looked like this, the weeds. The darker colors, the reds and the oranges, all the way up to almost 100% coverage. Only the blue areas had open water. So we wanted to affect a removal of that macrophyte population. And of course, part of that is removing the sediments. So in 2017, again, this is what the profile looked like. By 2022, the depth profile had been improved so that you had much better depth profiles around those margins. And taking a transect across that lake again, flipping it on its side, showing the purple, the before, and the black after, you've improved that depth profile from about 2 feet down to about 6 to 8 feet in much of that lake. And that's a vast improvement in the reduction of sediments. Back to that macrophyte profile in 2017. This is what the macrophyte profile looked like in 2022 with that improvement in the depth. And then the macrophytes also were much reduced, allowing recreation and allowing a lot more water volume. So the 5-foot contour line in 2017 looked like this. That 5-foot contour line in 2022 looked like this. And some of the numbers associated with that, the average depth in 2017, 5. By October of 22, improved to 8 for an increase of about 3 feet. And water volume, you looked at cubic yards of water volume, 250,000 in October, about 400, almost 400,000 in October of 2022, for an improvement of about 140,000, or an improvement of 55% in terms of water volume. Along with that is a reduction in those vegetation biovolumes went from 33% in October of 17 to 3.5% in October of 2022. So overall, a very good example of sediment decomposition and improvement of water volume and improvement in vegetation coverage. So these two examples then eliminate hypoxia, deplete sediment nutrient stockpile. Now we want to show how those two steps can help restore phytoplankton balance. In other words, the good algae or the beneficial algae. Let's move back to Indian Lake. Keep in mind, this was the example I used for the hypoxia. Now we want to look at how to improve the algae. Again, just to refresh, these were the profiles for oxygen, phosphorus, and ammonia from 2021 to 2023. And Indian Lake, again, looks like this. Restoring the phytoplankton balance. This is a percent taxa profile following those methods. Notice on the left, the darker green is the total number of algae. The lighter green is the algae and diatoms, which is what we want to increase. And the black and the blue are the cyanobacteria. And notice that in the beginning, there were quite a number of algae, but they were composed largely of the toxin-producing, non-producing cyanobacteria. Okay, looking at the taxa again, the darker green, the total, and the improvement in that total number to the June of 2023, May 2023, and 2024, and the improvement in the good algae during that same time period. The cyanobacteria were maintained at about the same level throughout that. So essentially, diversity was much improved, and the diversity was in the direction of the beneficial algae. Expanding that to look at the biovolume, not just the numbers, notice on the left that the total number of algae were composed almost entirely of cyanobacteria. And over the length of the program, it was improved. But let's expand that from 147 to 40 and expand that scale just to see how the improvement really looks. So on the left, 2020-22, most of those total numbers were of cyanobacteria, but notice in the treatment process how that has been reversed and that most of the improvement has been in the area of algae and diatoms. Along with that improvement in the good algae, green algae and diatoms, is an improvement in the zooplankton. In this case, total number of animals, arthropods and rotifers, arthropods being much larger, they're the insects. But notice on the left what the situation was in May of 2022 and how that improved over the course of the treatment process. Same thing with the zooplankton biomass. In this case, you notice that the arthropods compose most of that biomass, but they're the larger ones, the insects. They tend to be the ones that consume the rotifers and the protozoa. So back to that biovolume graph. Let's superimpose the zooplankton on that graph, showing you how the improvement in the diversity of the plankton and the phytoplankton, especially green algae and diatoms, has affected an improvement in the zooplankton numbers as well. So that diversity then sets up the stage to actually improve the whole system in terms of biodiversity, including fish back in the system. So we've done eliminating hypoxia, depleting sediment stockpiles, restoring phytoplankton balance, beneficial algae. Restoring the food web, restoring the nutrient clearance channels. And a quick example of that, this is Talvaca Reservoir in Puerto Rico. I won't go into the details at how that process was affected. Just keep in mind that that hypoxia reduction and the nutrient reduction allowed then a restoration of the food web, and the fish kills were stopped, fish populations restored, bird populations were improved. In other words, food web restoration, which then leads to nutrient clearance channels, fish going out of the system, for example, instead of nutrients being recycled in the system. A final quick example, one that I personally saw in Bowling Green, in Reservoir in Bowling Green, Ohio. The unique situation here, and this leads us back to the problems with Lake Erie. On the left, you see the Maumee River, kind of a chocolate, almost a chocolate brown, has a high level of cyanobacteria and other plankton. And of course, those nutrients and the plankton, phytoplankton, go into the western basin of Lake Erie. So Bowling Green is tasked then with having to utilize that water and treat it and produce potable water for the city of Bowling Green. So they pump water out of the rivers into the reservoir. And the challenge then is to reduce those population of cyanobacteria. So in 2016 versus 17, I'll show you the results. The program commenced April of 17. In August of 2016, the phytoplankton and the phycological count was 150,000 in the river, and in the reservoir, 124,000. 68% of that was cyanobacteria in the river, and 69% of that was cyanobacteria in the reservoir. That was unacceptable. Following treatment, July of 17, the total phycological count, July of 2017, 160,000 in the river, and in the reservoir reduced to 41,000. Cyanobacteria went from 42% in the river in 2017 to less than 1%. Well, in summary, and this is what I'd like to emphasize, how do we affect remediation and prevention? Well, it's a multi-step process, and it's a multi-step process that uses physical, chemical, biological methods, but they combine it, and it's done in a more effective, thoughtful, scientific manner. And it really comes down to, in the beginning, understanding cyanobacteria's competitive advantage and allowing systems theory to help you understand that so that you can better manage the biological ecosystems. Study the individual water body, design custom prevention and remediation program. Don't just put chemicals in, don't just put in aerators. Think about the design and placement of the aeration systems and how you then approach it with chemical and biological. So you want to eliminate hypoxia, deplete the sediment nutrient stockpiles, restore phytoplankton balance, in other words, beneficial algae like diatoms and green algae, not just them, but keep the cyanobacteria down or at a level that allows a much greater diversity. And that allows then a restoration of the food web to restore nutrient clearance. Again, keep in mind that this aeration, decomposition, sediment profiles is really just comes down to biodegradability degradable organic sediments. And all lakes tend to have some of this, but over the last century or more, lakes have generated a massive amount of those due to eutrophication from agriculture, development, human habitation. And these sediments are susceptible to aerobic digestion. It's biological. This is a principal method in thousands of activated sludge waste treatment plants, which oxidize the massive human organic waste being applied to a reservoir or a lake. And these multi-level methods have been successful in other places, Michigan, Pennsylvania, New York, and so on, and can be applied worldwide. So finally, I'd like to acknowledge again these examples of case studies, which I think have done a good job of showing you how those methods can be employed and employed in a thoughtful way. So this is SIS Bio, again, across the river in Pennsylvania. Thank you for your attention. That's my last slide, except for some key references. These are all references from agencies and reports. And I guess the point I'd like to make is that reading any of these, they're very good at talking about monitoring, detection, sort of understanding why blooms happen, but don't really do an effective job of reflecting on prevention and remediation and how that might be attained long term. Note at the bottom that even New Jersey has their freshwater response reports yearly. Utilize this information, but keep in mind that it's not the end all, that you're really out there to make long-term improvements to water quality and prevention of cyanobacteria, harmful algal blooms. Thank you.